DNA replication is a process by which DNA makes a copy of itself during cell division. Let's start with some vocab. Antiparallel means parts of a molecule having opposing direction. Origin of replication is a DNA sequence at which helicase unwinds to DNA double helix and DNA polymerase binds to initiate DNA replication. Helicase is an enzyme that unzips the DNA double helix. Primase, is an enzyme that helps to create a starting point for DNA replication. The replication fork is a point formed by the unwinding of a double helix. It's where DNA molecule is replicating. That's out of frame. DNA polymerase 2, its job is to make DNA from nucleotides. The leading strand is made continuously in the 5' to 3' direction. The lagging strand is also made in the 5' to 3 direction, but is made of Okazaki fragments and is made discontinuously. Okazaki fragments are newly formed DNA segments that make up the lagging strand and are later linked by DNA ligase. DNA ligase is like the glue for DNA and binds everything together. And semi-conservative is a method of replication which results in one new strand and one old strand. Initiation. The first step in... Oh my god. Oh no. Miss Torres, you need to let us edit this. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together. The separation of the two strands of the DNA creates a Y shape called the replication fork. This is also another picture of the replication fork in case this one wasn't clear enough. And that's the helicase unzipping the double helix. The two separated strands... the separation of the two single... I already read that. The two separated strands, which act as templates for making the new strands of DNA. One of the strands is oriented to the 3' to 5' direction towards the replication fork. This is the leading strand. The other strand is oriented in the 5' to 3' direction, away from the replication fork. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. Oh my gosh. Come on, Miss Torres. We need to be able to edit this. This is a whole mess. Also, in initiation, there is... Dang it, I forgot what I was going to say. Never mind. An elongation, the leading strand, which is on this side, is made continuously, and numerous DNA primers are made by the primate. Oh wait, I'm reading the wrong one. The leading strand is a short piece of RNA created a primer, which is produced by an enzyme called primase. I wrote that right here. That's their primer. Comes along and binds to the end of the leading strand, right there. The primase acts as a starting point for DNA synthesis. DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide bases to the strand of DNA in the 5' to 3' direction, which in this sort of replication is continuous. You see, I already connected them all together, but let's pretend that he's walking along it and matching up all the pairs. Also, this dark blue strand represents the new strand, and these light blue ones right here and right here are the old strands, and the same thing goes for this one over here. The lagging strand, numerous DNA primers are made by the primase enzyme and bind at various points along the lagging strand, which is right here, right here, right here, and right here. These are not for real. I just drew them on there because I needed to draw them on there. Chunks of DNA, which are these little lines in between the dots, are called Okazaki fragments and are then added to the lagging strand, also in the 5' to 3' direction. So it is going to go this way. Perfect. Also, in the initiation part, I drew a fork which better describes the direction of which the DNA is being replicated. And let's get back to this one. And in the lagging strand, this type of replication is discontinuous, as the Okazaki fragments will need to be joined up later. Right here, I drew them being connected with DNA ligase, which is like the glue, and bonds everything together in DNA replication. Lastly, we have termination, which is basically the end of replication and results in two molecules of DNA from this one. And let's see, Miss Torres. This is too much without some editing. Alright, we're back and we're better. Once all the bases are matched up, an enzyme called exonucleocase strips away the primers. So basically what it does is it takes away like this little one and this one, these ones over here. The gaps where the primers are then filled by more complementary nucleotides. The new strand is proofread and to make sure that there are no mistakes in the DNA sequence. Finally, an enzyme called DNA ligase, which is over here. I drew this one over here because I thought it showed a better representation of like how it forms, you know. And also I drew this upside down. I don't know. I wasn't thinking really, but yeah, it's definitely upside down. And it seals up the sequence of DNA into two continuous double strands, which are these two over here. The result of DNA replication is two DNA molecules consisting of one new. So one new is this dark blue, and one old chain, which is this lighter blue color. This is our original one, and this is what it's transformed into. And the reason why there's one new strand and one old strand is because this is a semi-conservative process, which means half of the chain is part of the original DNA molecule and half is brand new. Following replication, the new DNA automatically winds up into a double helix. So over here in this one, they're not like wound up. I just didn't know how to draw this better. But basically, they are wound up, and this is my double helix that's twisted up, but it's definitely more tightly wound in real life. And I think that's the end of this video.